is the age of induced pluripotent stem cells. So you'll hear a lot of research in the media as well as in your science journals on the development of this technology. And so just to summarize again, what we discussed in discussion today is that you can take viruses that express these four factors, c-MYC, KLF4, OCT4, and SOX2, and you can use these factors to differentiate fibroblasts into more pluripotent type of stem cells. And then these iPS cells can then be redifferentiated into specific cell types. Okay, and that these genes that are in the viruses are involved in normal biological processes. So, for example, c-MYC is important for, or is expressed in immortalized cells or tumor-like cells. iPS cells and embryonic stem cells are highly proliferative, so it makes sense that they would express c-MYC. KLF4 blocks apoptosis, which also promotes proliferation. And then OCT4 and SOX2 are transcription factors that are usually expressed in pluripotent stem cells. So we've talked about today in discussion how these four factors can change adult fibroblasts into a more embryonic stem cell type looking cell. Okay, so we talked a little bit about the low efficiency of this process, and this diagram shows, demonstrates that from the fibroblast. So here is a fibroblast at day 0, and really it takes about 30 days to get those very few iPS colonies. So it takes time for the reprogramming to occur, and it takes time for expression of genes that are usually associated with pluripotency to be activated. So first you start to see expression of a gene called alkaline phosphatase that's expressed mainly in pluripotent stem cells, but is also expressed in some differentiated cells. There's a protein antigen on the cell surface called SSEA1 that is highly expressed on embryonic stem cells, and that also starts to be expressed after 10 days. And then OCT4 and NANOG are transcription factors that are important for, or that are expressed in pluripotent stem cells. And all four of these markers are usually present in the iPS cells that you see at 30 days. So you can imagine if you're a patient and you have a disease that's not going to let you last 30 days, iPS is not going to help you, right? Because you need to do it faster. And so, so there's a lot of research going on now to try and increase the efficiency of induced pluripotency. The other molecular biology that is being investigated to try and improve iPS technology is to understand the epigenetics that occurs upon reprogramming. And so epigenetics is basically a big word for DNA methylation and histone modification. So it's basically changing the structure of the gene so that the gene expression is either there or not there. And so we talked about how how reprogramming can occur. We talked about how to look at DNA methylation and how to look at histone modifications in the iPS cells to see if they're really looking like embryonic stem cells. But if you think about it, some of these things have to happen stochastically, or that is, they have to happen randomly in the cells. So in addition to kind of using the science, there's a lot of luck involved in making a single iPS cell from a fibroblast. And so, and this also takes time. This reprogramming process seems to take time. So there's a lot of different research going on to try and control or make this epigenetic changes less random and more direct. So, for example, a lot of drug companies are looking for small molecules that can be used, cultured on fibroblasts, and try to reprogram them, so that way we won't have to use viruses that contain oncogenes. So trying to do it chemically instead of using natural genes. Okay. And so this is just a diagram to show, for example, the different epigenetics that can occur. For example, here, this is what I wanted to show you here. So in fibroblasts, there's a certain set of histone and DNA modifications. This represents a histone that is histone 3 that's methylated on lysine K9. And then in a partially reprogrammed fibroblast, it's been observed that some of the histones are no longer methylated on lysine 9, rather they are methylated on lysine 4, which makes their chromatin more open. And then iPS cells tend to have a different pattern, or fully reprogrammed iPS cells have a different pattern of histone and DNA modifications. But this happens sequentially. Okay, it doesn't happen instantly, which may contribute to the time that it takes for a fibroblast to be reprogrammed into a true iPS clone. Okay. So the cells that we talked about in the paper today were started out as mouse embryonic fibroblasts and adult tail-tip fibroblasts. So since that initial report, everybody's taking their somatic cell of choice and trying to turn it into an induced pluripotent stem cell. And so, for example, here they've made iPS from adult mouse liver and stomach cells, and these are the iPS lines that are created from those somatic cells, and they look very similar to the iPS clones that were made from the different fibroblasts. In addition, iPS cells have now been made from human fibroblasts as well. And so hematopoietic stem cells and cord blood cells have been also reprogrammed into induced pluripotent stem cells. And interestingly, cord blood cells, right, these are the cells that are collected when a baby's born and can be used for transplantation later if that person needs, say, a blood transfusion from themselves. So these are pretty early stage developmental cells, and they don't require all 4 factors to be reprogrammed. They actually only need 2. So that means that, or that suggests that the more stem-like of a cell you start out with may be more easily reprogrammed into an iPS cell. So what else can we use iPS cells for? So the fact that human iPS can occur has opened up a whole area of research into human disease. So currently there's a lot of mouse models of different diseases that exist that are used to test different hypotheses on how the human disease might work. So for example, there's models of multiple sclerosis in mice, there's models of diabetes in mice, but nobody really knows how exact those mouse models mimic the human model. And obviously we don't make human models of disease, right? So the models that we study are actually the people that have the disease. You can't go back and clone them. But what you can do now with iPS is that you can take, say, a skin cell from a person that has multiple sclerosis and reprogram it to an iPS-like state and kind of study how it develops into a neural cell and see, for example, if there's